start recording. Recording, okay. It should record. Let's see. Yeah, does it? Does it record? Yeah, okay, the blue bar is there. And now let's go over, share screen, start broadcast. And we are on the right channel, just for a change. That's a good thing. Good. And head over to our topic today. Okay, big question as per usual. Can you see my screen? Is the screen visible? Lovely. Thank you very much. Excellent. Good. So, and there's no attendance monitoring today because it is part of this enhancement week thingy. And I thought it might be of some interest because what I'm trying to do today is really link things together and also some stuff from other modules to give you a flavor of how tricky the design of pharmaceutical drugs, and I'm only talking about pharmaceutically relevant drugs. So I'm not talking about the stuff that people take for recreational purposes. Although, again, you know, the principle is exactly the same. And no, it's not in any way assessed or will be asked in any tests or exams or anything like that. This is just, how shall I say? What on earth is the seagull doing? Holy cow, this is not right. Hang on, sorry, I got slightly distracted. I'm just seeing a seagull that is trying to do either yoga or is pissed because it's trying to stand on its head. Oh, that's weird. Oh, yeah, it's gone. Sorry about that. It's just a session for fun and for, you know, for information. There is you don't have to take any notes if you don't want to do that, but I just thought this might be of interest. Okay, so let's get started because it's quite a bit of material that I want to go through with you today. And as I previously, when we were talking about inhibitors, as I started to say, drug design is at the core of the entire pharmaceutical industry, because that is what people do. They are trying to find drugs to combat diseases, to combat illnesses, and to, in general, to improve our lives. And how do you actually find such a drug? And let's not forget, drug design is really hard work. And here's a picture of what this looks like. So here's the workforce. And they're really working currently extremely hard. Just to give you a sort of a ballpark figure, from the day of inception, so when you have, as a scientist, you have the idea, I want to find a new drug that addresses some problems, to the day when this drug can actually be prescribed, we are very often talking about a period of, if it is a drug in humans, we are talking about sometimes up to 10 years development time. And we are also talking about sometimes costs in the range of easily $1 billion for the development of a drug. So if you are a company involved in drug design, you really need to make sure that you get it right, because no company can afford just to simply waste 10 years of development and $1 billion US dollars in development costs. So you need to get it right. And one of the things that people need to be absolutely clear about is they need to find a good target. Finding target. Finding a good target for the drug. What does that mean? Well, let's say we want to develop, let's say, a new antibiotic, or we want to develop a new painkiller or something like that. So we need to find an enzyme against which we develop this particular drug. We want to find a pathway that is a good target for this drug that we want to develop. And we want to make sure that we are very, very clear what we actually want to achieve. And that's very often, it sounds trivial, but that very often is a big question. If we want to find a new antibiotic, we need to be clear. What are the, for example, the bacteria against we want to find an antibiotic, and what would we like to target? What process we would like to target? Now, with antibiotics, sometimes it's not terribly difficult, actually, because luckily, microorganisms against which we want to develop a drug have very often very different metabolic processes compared to, let's say, humans. So in order to find a good target, we want the target to be specific. So specific. That means, ideally, when we develop a new antimicrobial drug, it should ideally kill the pathogen, but not the host. That is what we realistically want to achieve. So if it kills the host, it probably would also kill the pathogen, but then, you know, it's not terribly good if you kill your customers. So you want to avoid that. So you want to find a good target against which you can develop this particular drug that you then want to market, that you want to develop and then eventually market and recoup all the expenses that you had during the development process. So how do you find a target? Well, very often you already have a sort of a metabolic process in mind, where you say, right, this is a metabolic process that for the pathogen is absolutely essential, but does not happen to that extent or in that case in my host, in the human. So sometimes you can even find an enzyme that you want to target for your drugs. So finding, let's say, we want to find a good, and good here means something that is specific for the pathogen, finding a good enzyme. And now, once we have this enzyme, ideally this would be an enzyme that only is present in the pathogen, finding a good enzyme. Once we've got that, we can use our knowledge of inhibitors and try to find something that blocks the enzyme. So finding the enzyme, knowledge of inhibitors, of enzyme inhibitors, and we can then use these inhibitors to, come up with a drug that blocks this enzyme specifically in the pathogen, but ideally not in the host. And the most common form of inhibitors that we would probably encounter that the pharmaceutical industry is developing are inhibitors that mimic, that look like the active site, that look like the substrate. So these inhibitors are designed to bind to the active site. The inhibitor and the true substrate are mutually exclusive. So when the inhibitor binds, the substrate can no longer bind. What kind of inhibitors am I talking about? Question over to the audience. What inhibitors are we talking about? Absolutely right, guys. Yes, you truly understand the concept. Yes, we are looking at competitive inhibitors. We are looking at inhibitors that mutually exclusive to the substrate only bind to the free enzyme. And in this case, we are looking for active site inhibitors. Absolutely right. Now, of course, what we want to do is we want to develop this inhibitor so that they can fit nicely into the active site. But wait a moment, we have a problem here, don't we? We have an enzyme, we have a rough idea what the inhibitor should look like, because we know in most cases what the substrate should look like. But where on earth in this protein is the active site? How can we find the active site of an enzyme? Any ideas how we can find that? How can we find the active site of an enzyme? Alignment. Cami, I like your idea. What do you mean by alignment? What do you align? You're absolutely on the right track, but what do you align? You are spot on. Although you probably would not necessarily use the genetic code, you would use just simply the amino acid sequence of the protein, and you would compare the amino acid sequence of similar proteins from similar species. The idea behind that is actually that the same mechanism of catalysis. So when we're looking at enzymes, each enzyme has a mechanism of catalysis. That is how it works. And the same mechanism of catalysis is probably highly conserved between species. So nature doesn't invent the wheel all the time, over and over again. It uses the same mechanism for a particular enzyme reaction. And the same mechanism uses the same amino acids. So what we can do, as Kami suggested, what we can do is we can compare different species that have that enzyme that we are interested in. We compare the amino acid sequence and we try to figure out which amino acids are highly conserved. So here, for example, is a not particularly good example. It is a protein that or an enzyme that I've been working with for the last 20 years or so. It's a protein in E. coli, which is a potential drug target, actually. And we find this protein only in E. coli-like bacteria, where it is fairly conserved. Now, what we can do is we can align the sequences of similar proteins from other species and do a nice alignment. So what you see here, wherever you see a star like this one here, we have a perfect match in the amino acid sequence. Here, we find some conservative mismatches. So, for example, here we've got a Q and a K, for example, in different species. Here, that means we have similar amino acid properties, but not identical ones. And here, for example, we have a region where it's sort of pretty much random. It doesn't matter what the sequence is. Now, in this case, it's a little bit tricky because they are so similar. So it's not terribly easy to find out which one is the active site. We can do a little bit more digging and compare some bacteria that are not so so closely related. So you need a bit of a knowledge of a phylogeny of how you classify the microbes. And for example, you find that the coliform bacteria here, they are all very similar. But if you compare, for example, one of the binding sites in this particular protein with another sort of relative, in this case, Yersinia pestis, this is the bacterium that causes the plague, you find that they are slightly less related and you find differences. They have the same protein, but you can sort of work then your way. If you include less similar proteins, you can figure then out what might be where might be the binding site, because in the binding site, you still have the same amino acids in the substrate binding site, in the active site. And ideally, what you end up with is a model of the enzyme that you are interested in. So here, luckily, we had a model for our protein, for this protein called SIR-A. SIR-A is a protein that is essential for E. coli to survive stationary phase. And so if you take out this protein, in E. coli will not survive stationary phase. They will grow up to a certain level, but then when they reach stationary phase in their growth, they just simply die. Whereas other bacteria, when they reach this sort of level in the growth curve, so you've got a growth curve, that's time, and here is growth. what you usually find with bacteria, you reach something like that, and this plateau phase, where the cells don't multiply anymore, but they are still alive. And in E. coli, that very often is the dangerous phase where they produce toxins. That is the stationary phase, where no further growth happens. SIR-A is important for this stationary phase. Now, luckily, we had already a structure, a three-dimensional structure of all the amino acids, or most amino acids, and we obtained, people created this structure, so we knew where the different amino acids are located. We also saw that our protein that we have is basically made up of 3.5 domains. And if you remember, in BI300, we discussed the concept of domains in proteins. These are really units in the amino acid sequence that can fold nicely into compact structures. So we found that we have these nice domain structures of this protein. Most proteins have these domains, have such domains. They are made up of roughly around 100, 150 amino acids, each domain, and they are independently folding units. So in our protein, in this SIR-A protein, we had these three structures, and we could sort of, with these alignments that Cami previously suggested, we could figure out where roughly the binding site for the substrate might be located. Now, in this particular enzyme, in this SIR-A protein, it's noteworthy to mention that the substrate for SIR-A is other proteins. So SIR-A will bind to other proteins in E. coli and will do something with them. And this is essential for survival in stationary phase. So we knew already that the substrate binding site in SIR-A, where the active site actually is, that this must be a fairly large region, because otherwise other proteins would not be able to bind just to a tiny hole in the enzyme. Does that make sense? Or do you have questions so far? Yeah. Makes sense. Excellent, good. So what we needed to do is we needed to figure out where in this three domain structure, or 3.5, we didn't count the C domain here, nobody knows what that really does. We needed to figure out where is the substrate binding site. And just by looking at it, we thought, well, actually, there seems to be an interesting area here. And this area is this one here. It's sort of a big, how shall I say, a big cleft or something. And that very often looks suspiciously like we could bind something there. And if you look at the amino acids in there, again, you can say, oh, look at that. There might be some interactions going on. And in fact, what you can do is you can use computer programs that actually predict substrate binding sites. And we used a program from the company Accelris That's a drug development program. And we tried to figure out where is this substrate binding site. And what you can see here with these neon green things, the program actually predicted where a substrate binding site is potentially located. But this is only a prediction. So we had to do some experiments, we had to verify it. And one way of verifying it is you just simply chop off parts of this SIRA molecule and see whether it still binds. And if it does bind, then you've chopped off the wrong part because then the substrate binding site is still present. So we found out that we have a pretty good potential binding site. Unfortunately, we also found out that apparently there seems to be another binding site in this region, but let's not worry about that. This is just this SIRA because it's an awkward protein to work with. But hey, we wanted some challenge. So we had a good idea where this substrate binding site is, and now we wanted to see, can we come up with something that might block this binding site, that is a competitive inhibitor for this binding site, that binds to this substrate binding site, to this active site, and blocks it, so that the true substrate no longer can bind. Luckily, we had already a sort of a model substrate. This is our model substrate here. Model substrate. So we had a good idea what can fit into this binding site. So what we did was, and this is a short peptide. I can't remember. I think it's made up of 14 amino acids. 14 amino acids. That is the model substrate that we used, and we knew that this model substrate actually binds quite well into the binding site of SIRA, into the active site of SIRA. And one way you can check that is if you've got the correct substrate of SIRA, then you can add this, and SIRA will do something with the correct substrate. It will catalyze a reaction. So that's the normal process. So we had the enzyme plus the substrate, and it did something, enzyme plus product. But now what we did was what we've discussed with the competitive inhibitors. We used our model peptide, and used this as an inhibitor. So we formed an enzyme model peptide complex. And we no longer got the product out. And we looked at this model peptide. As I said, it's 14 amino acids long, so it's not terribly complicated. But we discovered that it sort of needs to be a sort of twisted. And you see this twist here with some residues sticking out. We looked at it, and we could sort of model this peptide and say, right, what does it look like when it actually binds? So here we use this model peptide, and we tried to put that in the computer into the structure of SIRA, into the active site of SIRA. And you see, for example, here, this large green thing. This is from SIRA. What is this green thing? What would you describe it? It's a secondary structure, or what would this be? Bingo! It's an alpha helix. Absolutely right. Yeah. So we saw there is an alpha helix that really is close to our peptide. So we've got phenylalanine of the model peptide, tryptophan of the model peptide, and we could see actually the structures of SIR-A which are in close proximity. So here we've got an alpha helix. Here we've got a sort of a bit of an unordered structure, which indicates an unordered structure seems to be quite give the area of some flexibility. And this then moves on into another helix here. So we already then at that point had an idea of what is the area surrounding our model peptide. So what does the structure of the binding site potentially look like? And then we carried on with that. We used a technology which is called STD. NMR has nothing to do with sexually transmitted diseases. It actually is a method that measures which particular parts of our model peptide, and that's our model peptide here, which particular parts of the model peptide are in close contact with amino acids from the substrate binding site. And everything that is red is very close contact with amino acids. So you see here these amino acids in this phenylalanine ring here. These hydrogen atoms are in very close contact with amino acids from SIR-A, from the binding site. Again, these here are in close contact. Here we've got only one in close contact. The neighboring two hydrogens are not in close contact. So what we can identify is particular properties of our model peptide, of the inhibitor, that is in close contact with amino acids from the substrate binding site. And what we can do now, and what people do now, is we can employ the tools of what is called Chemo Informatics. We have an idea of what the inhibitor should look like. We know that there needs to be, for example, an aromatic ring with some hydrogens attached to it that is in close contact with amino acids from the substrate binding site. And with Chemo Informatics, we can actually describe chemical compounds in a way we can then store this information in a big database. So currently we've got about, I think, 2.5 million chemical compounds cataloged in a database. And this database needs to have a sort of a common language in a way that I call the SMILES, where you write the particular properties of a chemical compound in a way that a computer can read that. So I don't speak SMILES, but Chemo Informatics actually has stored the critical chemical features of loads and loads of target molecules. And once you've got this, you translate this molecule, and there are computer programs that translate potential molecules into this SMILES notation. You can store it, and then you can search this database. And again, you can check out if there are what the chemical properties are of these potential interesting compounds. So here is another database, this more inspiration database, where you put in a molecule and it translates it into a smile, and it comes up with interesting properties that this molecule has. For example, the volume of this molecule. And the volume is really important because if you make your inhibitor too big, then it can't reach the active site. So what we now could do is we could ask the computer to come up with suggestions for what is called leads, potential lead molecules, potential inhibitors that fulfill the criteria that we specify. We say, for example, we need aromatic ring in this particular orientation. This aromatic ring should be connected then to something else that is not aromatic but positively charged, for example. And then there should be another aromatic ring. And then the computer goes through this Chemo Informatics database and says, hey, I've got 15 compounds for you that fulfill these specifications that you gave me that might be good candidates for your particular purpose. And what we then do is, since we have the structure of the binding site, the three-dimensional amino acid structure of the binding site, what we can do is we can use yet another program where we model our potential inhibitor, our lead, with all the correct information, with all the correct specifications. There is an aromatic ring, there is then a positively charged residue close to it, and there is another aromatic ring, but it has to have a slightly kinked conformation. We then can take this molecule and perform what is called in silico docking, where we put this molecule here, this solid molecule, our inhibitor, into the active site, and we see then, does it really bind nicely? Does it make contact with the right amino acids in the active site? Or is it probably shaped in an awkward position that it's not binding terribly well, or something like that? So we can look at the interaction map between our potential lead, our ligand, if you like, our inhibitor, and the active site in our enzyme, in our target enzyme. And what we can do then is say, right, out of these 20 potential leads, we found 10 of these compounds that are really, really interesting. So let's carry out further analysis with these leads. And for example, what we need to do then is, if we've got some interesting leads, let's say we have five potential leads, where we think that they are really good, strong inhibitors. Strong inhibitors. We now take these potential inhibitors, our potential drugs that we are going to market, and now we are doing experiments with them. Before that, we just did the whole thing in the computer. That is why this method is called rational drug design. Rational because we had a very good understanding of what our target will look like, and we could do everything in silico in the computer. And now we have 5 potential leads that we think are good inhibitors. Now we need to do some experiments with them. Which one really is the best? Of course, it could be that we are barking up totally the wrong tree, and none of them actually shows an inhibitory effect, which would be a pain. But luckily, we haven't spent too much time because everything is done in the computer, and we haven't wasted too much resources and chemicals because we have not done yet massive experiments on that. Of course, once we have good leads, we also need to make sure that they are not just great binding, great inhibitors that block the enzyme. We also need to make sure that these candidates, that's another word for lead, are suitable as drugs. So if we develop a candidate which is a very, very good inhibitor, but if it kills the host, if it kills the patient who we want to treat, then it's no good. So we need to identify what is the toxicity of this compound. What is how quickly is it removed from the system? Removed from organisms. So the clearance, for example. Now, if a particular candidate is found to be very, very toxic, then obviously we can't use it. But what we can do is we can take this candidate that is so toxic, we can try to modify it. So, for example, to reduce the toxicity for the host, for the patient, we can add some other chemical groups to it, which makes it less toxic. We can modify it and we feed it back into the docking process and say, well, does it still bind very well? And is it now less toxic? And if it is less toxic, oh, let's develop this further. So, for example, if you do cancer treatment, you would, for example, use a very, very highly toxic substance, like, for example, cisplatinum. This is extremely toxic, but it's more toxic for the cancer cells than for normal cells. So it will kill normal cells, but cancer cells suffer even more. However, in this case, the toxicity is something that you want, but you don't want to make it too toxic because then the patient will die. So all these tests, toxicity, does it affect cell survival, for example? You don't want the drug to be good to the pathogen that you're treating, but you want to be the drug not to be too detrimental to the host. So you need to check what are the modifications that you can do to make it less toxic, but still potent enough. And again, there are several tests that you can do, and there are certain criteria that you can use for these candidates. And just in case you wonder, this is exactly what you are currently doing in the BI301 in the skills module, skills for bioscientists. You are testing a series of candidates in the assessed practical. So you are testing for, does this drug actually kill cells? That is what you are doing in the BI301 assessed practicals at the moment. You have a number of potential candidates that went through this screening process, and now you are trying to figure out which one is probably even better. You are, in a way, doing these testing for us to see which one really, which drug we should take forward. Does that make sense so far? Are you happy with that? Or do you have any questions? Now, there is a catch to it, as you might expect. And that catch relies on your knowledge and understanding of inhibitors. So let's say we have a pathway, A, B, C, D, something like that. And we decide that we inhibit the enzyme that catalyzes the reaction between B and C. So this is our drug that we've developed. And this drug is a competitive inhibitor. Right. So far, so good. We found our drug. We found a good competitive inhibitor. Now, tell me, what will happen if we add this drug to the pathway? What will happen if we block the reaction from B to C? What happens to the concentration of C? What will happen to the concentration of C? That's not a trick question. You're absolutely right. It goes down. Totally right. What happens to the concentration of B? So now I'm asking for B. What happens to the concentration of B in this pathway? Absolutely right. The concentration of B goes up. Now, B, of course, is a substrate for the enzyme that we inhibit. So B concentration goes up. Now tell me, under which conditions does a competitive inhibitor work best? Competitive inhibitors work best when the substrate concentration is, what would you put in? Competitive inhibitors work best when the substrate concentration is, you nailed it, totally, is very low. If the substrate concentration is low, then the inhibitor works. If the substrate concentration is low, then our drug works very well. But in this case, by adding the drug, you see what happens here. And B is our substrate here. The substrate concentration goes up. Do you see the problem? We add the inhibitor, the substrate concentration goes up, which basically renders the inhibitor useless because the substrate concentration goes up. Oh shoot, $1 billion just wasted. Actually, it's not that bad because remember, most pathways actually have branches. They are not just linear. We might direct this pathway into another direction. So we force the pathway, instead of going from C to D to God knows where, we force the pathway into B, E, F, and so on and so forth. That means our concentration of B will not go up too much, and we have a nice block here. So all is not lost. If we have a branch point here in the pathway, then we might be able to use this drug. And even better, maybe we can use this drug to develop an even better inhibitor. Because what would be a really good inhibitor if the substrate concentration was very high? Which inhibitor is very good at blocking the enzyme when the substrate concentration is high? Which inhibitor is that? What kind of inhibitor is good at doing the job when the substrate concentration is very high? Absolutely right. An uncompetitive inhibitor is really good in this case. But how do we find an uncompetitive inhibitor? Remember, uncompetitive inhibitor, the substrate needs to bind first to the active site. This introduces a conformational change, and then, only then, the site for the inhibitor will be accessible somewhere else. We don't know where else. But this is only a very short period of time. This only happens while the substrate, the true substrate, is actually bound to the enzyme. But with our competitive inhibitor, with our drug now, we have something that actually binds. This here now is our drug. That's the inhibitor. That's the drug that binds to the active site. And this active site, this drug, stays longer, much longer in the active site because it blocks it. Now, what this binding event now does is, of course, it does this conformational change with the drug bound, not the substrate, with the drug bound. And now we can look at the structure. We can look at the structure of this protein again, of our target again. We can try to get the three-dimensional structure. We compare the two structures. This is the enzyme, just shortly after the drug has bound, and this is the new structure. And now we compare the structures of these two and find that the enzyme has changed quite a bit. We try to find now, with the tools that I described, a binding site for our second. This is the second drug that we can develop with that. And that then is an uncompetitive inhibitor. And this uncompetitive inhibitor would then bind even if the substrate concentrations are very, very high, even if our B was very high. So what we can do is, with our first inhibitor, with the drug that we first developed, this competitive inhibitor, we use this to develop a second generation drug that no longer acts as a competitive inhibitor, but now acts as an uncompetitive inhibitor. And we have then basically two drugs that work both under high and low substrate concentrations. My time is up. I hope this gave you a little bit of an insight into how drug development is carried out. And that is basically what all the drug companies are doing. Time is up. If you've got any questions, please fire away. But otherwise, thank you very much for watching. And I shall see you next week for the next BI301. But tomorrow we've got a BI308 lecture about more statistics. So thank you very much and enjoy your day. Be good. Don't do anything I wouldn't do. Take care. Bye-bye.